So in the second half here, we're going to look at expiration, control, and transport. All right, the force that's responsible for normal expiration comes from elastic recoil of tissues and surface tension. Lungs have lots of elastic tissue, which allows for stretching and expanding, which is important. The recoil of those elastic fibers in lung tissue reduces pressure in the pleural cavity. So now these factors are going to increase the intra-alveolar pressure about 1 millimeter per mercury above atmospheric pressure. So I now have a slight increased pressure inside, slightly lower outside, and then I can have forced expiration when I have contraction of internal intercostal and the abdominal muscles. So let's break this down into events. The first event in expiration is the diaphragm and the external intercostal respiratory muscles are going to relax. So the diaphragm is going to relax. Elastic tissues are stretched, they recoil, and then surface tension is going to pull in the alveolar walls. Tissues are going to recoil around the lungs, which increases the pressure, and then air is forced out of the lungs. So how do we measure how well the lungs are working? We do this by measuring and looking at respiratory air volumes and total capacity, the different capacities of the lung. Different degrees of effort in breathing move different volumes of air in and out of the lungs. And we know this instinctively, right? If I'm just sitting here and breathing, not thinking about it, it's hard to not think about it when you're thinking about it. You're just breathing in a small amount, but I can take in and force out a lot more air than I do when I'm just naturally breathing without any conscious thought. So the measurement of such air volumes is called spirometry. Spirometry, and it's done by a spirometer. So 3 distinct respiratory volumes can be measured using spirometry, and there is a fourth that we can't measure that way, and that's called residual volume. The volume of air that enters the lungs during forced maximal inspiration is the inspiratory reserve volume. So 1 inspiration plus expiration, that is going to be what we call a respiratory cycle. Tidal volume is the, and that one's down here, tidal volume. All right, what else did we say? Anyway, tidal volume is the volume of air entering or leaving during that cycle. For normal respiration, that is about 500 milliliters. Okay, 500 milliliters or 500 cc's. Those are equivalent units. During forced maximal inspiration, a volume of air in addition to the resting tidal volume is called the inspiratory reserve volume that enters the lungs, and it equals about 3,000 milliliters. 3,000 milliliters. So during a forced maximal forced expiration, about 1,100 milliliters of air in addition to the resting tidal volume can be expelled from the lungs. We call that the expiratory reserve volume. Okay, so even after the most forceful expiration, however, there's always about 1,200 milliliters of air remaining in the lungs, and we call that the residual volume because that's what resides still in the lungs. Okay, so that's a lot of terminology there. I would go and get all of these terms and write them down and look at the definitions so you understand them better. And you can listen to me tell them to you again as well. So 4 respiratory capacities can be calculated by combining 2 or more of the respiratory volumes. We've got the just a couple more definitions here. Not all the air from breathing reaches the alveoli. About 150 milliliters stays in the trachea, the bronchi, and the bronchioles. Gas is not exchanged here in these passages, and so we call that anatomic dead space. Anatomic dead space. Alveolar dead space is when portions of the alveoli are not functioning, and physiological dead space is a combination of the two above, anatomic and alveolar. These rates are important in determining the concentration of oxygen and carbon dioxide in alveoli. The volume of each of the new atmosphere moved into each respiratory passage per minute is what we call minute ventilation. That is calculated by multiplying tidal volume times breathing rate. So for example, if I had 500 milliliters for my tidal volume and a breathing rate of 12 breaths per minute, I would multiply those and get 6,000 milliliters per minute. There. So for each breath, well, let me back up for a second. Tidal volume then means that the physiological dead space is multiplied by the breathing rate, and we will call that the alveolar ventilation rate. That's the volume of air that actually reaches the alveoli, alveolar ventilation rate. It's really more important that you understand what these are than how to calculate them. Tidal volume breathing multiplied by the breathing rate, that's a good one, but anyway, so the reason this is important is this can actually impact the concentration of oxygen and carbon dioxide that is in the alveoli. Non-respiratory air movements are those that are anything besides breathing. This is coughing, sneezing, crying, and laughing, and these basically serve to clear the air passageways. Clearing our throat. Sneezing clears the upper respiratory passage. Hiccups are caused by inspiration due to diaphragm spasm. Breathing functions are served to deliver oxygen to cells where it takes part, as we said earlier, in cellular respiration. And that allows us to liberate energy from nutrient molecules and get rid of carbon dioxide, maintain that normal pH. Normal breathing is that rhythmic, involuntary act that continues when we are unconscious. We can sometimes also control them voluntarily, as in when we want to take a deep breath or hold our breath or exhale with extra. There are respiratory areas in the brainstem that control breathing. These are groups of neurons. We have impulses that travel on the cranial nerves, spinal nerves, and these cause inspiration and expiration. These respiratory areas also adjust the rate of depth of breathing to meet with cellular needs for oxygen and removal of carbon dioxide. So this is why when you are exercising, your breathing increases and you breathe deeper. The respiratory control areas are in the pons and the medulla. Two parts here are of special interest for us. The respiratory center of the medulla has two groups of neurons. Those run the length of the medulla oblongata. We have the ventral respiratory group and the dorsal respiratory group. The ventral respiratory group, we believe, controls basic rhythmic breathing, inspiration, and expiration. And it also kind of depends, of course, on those experimental conditions. The dorsal respiratory group stimulates inspiratory muscles, mainly the diaphragm, and this aids in processing of sensory information for the respiratory system, and it may also affect cardiopulmonary reflexes that affect the rhythm involved in breathing. Respiratory group in the pons contains neurons that contribute to rhythm of breathing by limiting the respiration. So that's the pontine respiratory group. Here we've got some different factors that affect breathing rate and depth. The partial pressures of the gases like oxygen and carbon dioxide. How much your lung can actually stretch. What is your emotional state and physical activity, which we've already discussed, okay? And so these types of things can affect your breathing rate. All right, so we also have some changes in concentrations in the blood of oxygen and carbon dioxide, the dissolved gases, and the pH of the blood that can actually stimulate some chemoreceptors and affect breathing. Motor impulses will then travel from the respiratory center all the way to the diaphragm and external intercostal muscles in response. Contraction of those muscles, of course, will cause the lungs to expand, and those are going to trigger what we call mechanoreceptors, and these can feel that stretching and respond to that stretching, and they send impulses back saying, hey, you've stretched far enough, don't go any further. And so it's an inhibitory impulse that goes back and says it's time to stop stretching so that we don't overinflate our lungs. Alveoli, this is a nice microscopic view here, are the sites of that gas exchange between air and the blood. The pores permit air to pass from one alveoli to another, and they provide alternate pathways. We have macrophages that actually help to keep the alveoli clean, and the cells of the alveoli secrete a surfactant. It's a type 2 surfactant, but it's a surfactant. So moisture in the pleural membrane helps to expand the lungs, but in the alveoli it does the opposite. Attraction of water actually creates surface tension, which makes it difficult to inflate the alveoli. So alveoli have surfactant to offset this. You're probably thinking, what is a surfactant? That is a substance that reduces surface tension of something. It can act as a detergent or an emulsifier or a dispersant. Okay, makes it disperse. And so this surfactant actually reduces the surface tension. So respiration is the entire process of gas exchange between the atmosphere and the cells. Gases diffuse from high to low concentration down their concentration gradient. We have different factors that can affect diffusion across the membrane. Things like surface area, having a shorter distance to travel, greater solubility of the gases, which means are they going to dissolve in water? And then is there a steeper pressure gradient? You can have diseases that harm the respiratory membrane, something like pneumonia, or reduce surface area for diffusion, like that's what emphysema does, and these can impair gas exchange, and that often calls for increased oxygen in order to treat. So this is showing you how those gases move along their concentration gradient. They're exchanged between alveolar air and capillary blood. So here we have a capillary that we are looking at, okay? We have a capillary and we can see that we have some dissolved carbon dioxide and we have some dissolved oxygen. And so we have carbon dioxide up here. It's in a higher concentration, if I can get the pen working, a higher concentration in the capillary, so it's going to naturally want to diffuse out to a lower concentration. And you can see that we have the opposite effect with oxygen. We have 104 millimeters of mercury here, which is much higher than the 40 millimeters of mercury in oxygen, and so it's going to diffuse in. Why is all this important to understand? Well, blood transports oxygen and carbon dioxide between the lungs and the body cells. As those gases enter the blood, then they dissolve in the plasma, combine with other atoms or molecules, and then they're sent all over the body to do their jobs. Primarily, oxygen is to be that final electron acceptor and allow us to make ATP through cellular respiration. So 98% of oxygen carried in the blood is bound to hemoglobin. And once it's bound to hemoglobin, we then call that oxyhemoglobin, okay? The rest is then dissolved in the blood plasma. Oxygen binds loosely to iron. That means it's easy to remove in the heme unit of your hemoglobin, okay? And so one hemoglobin can bind 4 oxygen molecules. So we have chemical bonds between oxygen and hemoglobin. I said they're weak, they're loose, so oxygen wants to leave. Factors that can increase us to release oxygen from hemoglobin are things like a decrease in the pressure of oxygen, an increase in carbon dioxide, an increase in acidity, and an increase in temperature. It is these mechanisms that allows oxygen to move from the blood into the tissues where it is needed. About 75% of the oxygen stays bound to the hemoglobin in the venous blood to be sure that we have safe carbon dioxide levels and keeping our pH at a level rate. Oxygen bound to iron in the hemoglobin is what gives blood the red color. The red color. When the pressure of oxygen decreases, the oxyhemoglobin is then going to release the oxygen molecules. Well, why do we care about that? Well, this is what happens in muscles. The tissues have used up oxygen in respiration, and we need a mechanism in order to trigger the body into delivering more oxygen. So pressure, a partial pressure increase of carbon dioxide, a change in acidity, and even temperature can increase how much oxygen is released. This helps the more oxygen leave the blood and enter the skeletal muscle when it's needed. So things like increases in muscular activity and using the oxygen cause that pressure of the carbon dioxide to increase and the pH to decrease, and that raises the temperature, which also affects this pressure. And so this triggers the oxyhemoglobin to release more oxygen and resupply the body. Okay, so carbon dioxide transport basically works the opposite way of oxygen. It's going the opposite direction. The gradient is opposite of that of oxygen, so the carbon dioxide flows down its gradient from high to low. So here we have a capillary. We're going to have a higher amount of carbon dioxide outside in the tissues than we do inside the blood, and so it is going to flow into the capillaries due to the concentration gradient. Now, the higher the partial pressure of carbon dioxide, the more carbon dioxide will go into solution, which would in this case would be blood. Carbon dioxide can bind to different amine groups. So oxygen and carbon dioxide don't have to compete for the same binding site on hemoglobin. That's important. Hemoglobin can carry both. When it is bound, the molecule is then called, when I'm talking about the carbon dioxide, is bound. It's called carbaminohemoglobin. This is also relevant because of the bicarbonate acid buffer system. We spoke earlier about how the respiratory system was involved in keeping that pH at a normal rate. So it was part of homeostasis. So if you look at this bicarbonate ion, the carbon dioxide is mostly transported as a bicarbonate ion. And so here at the bottom, you can see this reaction, okay, between carbon dioxide and oxygen. So we're able to remove that bicarbonate ion in the form of carbon dioxide and get rid of it. And so we're able to expel that from the body, which can alter the pH. And we're also able to hold on to it if we need to in order to alter the pH as well. Just as a side note, when you hyperventilate, that is accompanied, of course, by an increase in carbon dioxide and a rise in pH, and that is what triggers the symptoms that you feel due to hyperventilation. So changes that occur in the respiratory system that accompany agent are things like thickened mucus, we slow our swallowing reflex, the cilia become less active or even vanish, they disappear. If you're exposed to air pollution or smoking, you may develop chronic bronchitis, emphysema, or lung cancer. Your vital capacity will drop after the age of 40. Alveolar walls become thinner, so there's less surface area available for gas exchange. Your dead space increases. You can develop what's called a barrel chest, and so it does just get generally harder to breathe as you get older. A barrel chest is a condition in which the chest appears to be partially inflated all the time, with the rib cage broadened as if in the middle of a deep breath, and so the person finds it hard to breathe normally. We're also more susceptible to respiratory infections.